Okay, guys, so we've been talking about the physiology of the stomach, how the stomach works, and now let's keep on going with that idea. So we introduced this idea of peristalsis, which is these wave-like contractions of the smooth muscle in the digestive system. We keep that idea going now. So as our food moves through our stomach, these peristaltic contractions of the smooth muscles, they kind of like make a wave motion. And these wave motions will pass through our stomach every 15 to 25 seconds. Depending on how active the stomach is. So when you hear your stomach growling and everyone kind of says that you're hungry, well, that's not necessarily true. What's happening is those muscles are contracting and it's kind of making a bit of a squishy sound with your food. The reason why this happens in the stomach is because these waves will mix the food that we eat with all those secretions from the gastric glands, like we talked about, the gastric juice, and it's that highly acidic. It's got that those enzymes in there that we talked about, partially digested food that we've called chyme, and those peristaltic movements are going to mix all that stuff up and try and pass it through the rest of the digestive system. And what will happen is the chyme will be constantly being subject to this acid, to these digestive enzymes, and it'll start to break down our food into more useful chunks that our body is more readily able to use. So as these peristaltic waves pass through the stomach and they reach the pylorus, remember, which is the very bottom part of the stomach, these waves will push through small amounts of chyme into the small intestine from the stomach. Now this pushing through of the chyme into the first part of the small intestine, which you can see here, is called the duodenum or the duodenum, depending on who you talk to. This idea is called gastric emptying, and over time, of course, we'll empty the stomach contents, and it'll all pass through and move from the stomach into the small intestine. And the stomach acts as a place for mixing and initial breakdown, initial digestion, but most of the absorption that we're going to do is going to happen in the small intestine. So that's where we're going to take in most of the benefits from our food. So it's important to move this along. So every 15 to 25, 30 seconds, we're going to have about 3 milliliters of chyme pushed in through into your small intestine. And if you think about how big a volume that is, if you think about those little plastic droppers we have in the labs, way back when we could go into labs, well, one of those is about 2 milliliters. So you did 1.5 of those every 30 seconds or so. That's what's going to be emptied into your small intestine. And we can see, we have a look here about what's actually happening with this peristalsis. So you can see in numbers here 1 and 2, we've got the contractions of the muscles in the stomach, and that's going to force the chyme down into the bottom part of the stomach where it gets into the pylorus. Now every 15 to 25 seconds, it's going to push through our 3 milliliters of chyme. It's going to enter into the duodenum, which is the first part of the small intestine. Now when the pyloric sphincter opens, that's what allows the chyme to move into the small intestine. When that muscle closes, right here, number 5, what's going to happen is it's going to force that chyme back into the stomach, and then as that happens, it continues to get mixed up. So the mixing and churning continues in the stomach, and then when those muscle contractions happen again, it pushes another 3 milliliters of chyme through into the duodenum. Now, usually after 2 to 4 hours after eating, most of your food has been mixed up and it's been, initially digested or starting to become digested in the stomach, and it's been pushed into the small intestine. As a rule, generally, we can see foods that are rich in carbohydrates, so pasta, rice, breads. Those are going to be pushed through the stomach fastest. There's not much we can do in the stomach with those type of foods. So they basically get mixed up, broken down into smaller chunks, and then pushed through. The protein-rich foods, so meat, fish, those type of things, they're going to stay in the stomach a little bit longer, where we can have those protein enzymes like pepsin work on them. And our fat food, or fat-rich food, stuff with lots of lipids, that's going to take the longest time in the stomach. But generally, after 4 hours after a meal, that food's passed through the stomach, and it's mostly into the small intestine. In the stomach, we're going to have some digestion, of course, some chemical digestion. Now, the lipids and carbohydrates, the digestion is going to continue from the mouth, because remember, we started in the mouth by having the salivary amylase and the lingual lipase, and this digestion is going to continue even though it gets into the stomach where the pH, falls quite low. Those two initial enzymes, salivary amylase and lingual lipase, they're still going to work until we get to about pH 4.5. After we get down into a pH 4.5, we start to lose function. But if you look at lingual lipase, for example, it can work down to as low as 3 before it gets denatured and stops working. So the enzymes that started digestion of carbohydrates in your mouth will continue digesting lipids and carbohydrates in your stomach until the stomach acid denatures those enzymes, and then those reactions will stop. Digestion of proteins are the big thing that would happen in the stomach because we have this enzyme called pepsin that we talked about earlier. Pepsin activity is really going to pick up when we get to a pH that's lower than 2 or around 2. That's the optimum pH for pepsin, and so we've got the optimum temperature, which is going to be body temperature around 37 degrees Celsius, and then we also get the optimum pH around 1.5 or 2, and that's going to allow pepsin to work at its all-time best. Protein digestion is going to continue into the small intestine, but it won't be with pepsin because we're going to change the pH drastically once we move into the small intestine. Now, in the stomach, in terms of absorption, so we've broken down the food mechanically with all those stomach muscles. We've broken down the food chemically using the gastric juices and our enzymes. But the other part of digestion, the absorption, taking stuff from our food, that's really, really important. Not very much of that will happen in the stomach. The reason why is because we've got this lining that covers the stomach that protects it from the acid, protects it from the pepsin enzyme. That prevents the epithelial cells of the stomach from coming into direct contact with the chyme. So there's no real mechanism to get our food, from the lumen of the stomach across the cell membrane and into the tissue. Also, the epithelial cells in the stomach, they don't have those specialized transport mechanisms like we'll see when we get into our intestinal cells. Another thing, of course, when we're having stuff pass through membranes, we almost always need it to be dissolvable in water. It needs to be soluble, and the gastric lining is going to be impermeable to water. So it doesn't let a lot of water or any water move through it. So this stomach lining, the lining of the inside of the stomach, the gastric lining, as you can see it's called there, that's going to prevent most absorption from happening inside the stomach. So when our food leaves, or the chyme leaves the stomach, digestion is nowhere near complete. Okay, it's just started the journey that it's going to take as it moves through the rest of the digestive tract. Now, most absorption doesn't happen in the stomach, that's for sure, but there are things that the stomach does absorb. It will absorb some water. When it does that, it will move across some ions. We'll also see some short chain fatty acids, not big ones. And there are certain drugs that are absorbable in the stomach. Aspirin, for example, it gets absorbed in the stomach, and so does alcohol. But those are, it's a very short list of what can be absorbed in the stomach. Most of the absorption that we're going to take, the place where we get the most benefits from our food absorbed from the lumen and into our tissue, is going to happen in our small intestine. And that moves us on to the second part of this video, and we're going to start talking about the small intestine and the pancreas. Okay, so the first thing we need to do when we're talking about the small intestine is, of course, talk about some of the anatomy. Once we get some of the anatomy done, then we can move into the physiology, which is going to get a little bit more complicated. But when we're talking about the small intestine, the big function of the small intestine is going to be digestion and absorption. Okay, so 90% of the absorption that happens in the digestive system is going to happen here in the small intestine. The thing about it is, the tricky part, the mucosa, which is the inner tissue layer of the lining of the tissue wall of the small intestine, it only produces a few of the enzymes involved. Okay, so most of the enzymes that are released inside of the lumen of the small intestine that help with all of this digestion is not produced by the small intestine. So where do they come from? There are several accessory organs that are associated with the digestive system, and up until this point we haven't really talked about them. Now, what we consider an accessory organ is an organ that contributes to the digestive system, but food doesn't actually pass through it. Okay, so it's not part of the digestive tract, which is that tube that starts at the mouth, runs all the way through the body, and ends at the anus. What we have here are these organs that are connected to the digestive system, but the food doesn't actually pass through them. So the three major accessory organs that are associated with the small intestine are going to be the pancreas, and that's going to secrete digestive enzymes and buffers. That's going to help neutralize that acidic nature of the chyme so that we can have digestion at a different pH. The next big organ that we're going to talk about is the liver, and liver secretes a bile, which is made up of buffers and salts. And bile has a big role to play in the digestion and the absorption of lipids, so fats. And then finally, the gallbladder. The gallbladder acts as a storage organ, and that stores the products of the liver, which is the bile. So if you look over here on the picture, the stomach would be, in this area right here, somewhere along there, and you can see the pancreas, this organ, the light organ right below the stomach. The gallbladder here is connected to the liver using the common bile duct. So the liver will produce the bile. It'll get stored in the gallbladder and emptied into the duodenum, the small intestine down here. And as the pancreas will produce its enzymes and buffers, and that'll also get released into the small intestine. So those are the major accessory organs that are going to be responsible for most of the enzymes that contribute to the digestion that happens in the small intestine. So let's have a look at the lining of the intestinal wall, the small intestine, and just like in the stomach wall, the tissue design is going to be quite similar. On the inside, we're going to have the mucosa, the submucosa, the muscularis, and then, but on the outside, instead of the adventitia, we're going to have something called the serosa. Okay, it's the same idea. It's the outside part of the structure, but it's called a slightly different name. Another thing you're going to notice if you look at the small intestine is we see a lot of these little tiny detail, and we'll go into that right now, and I'll tell you why that happens. So here's a look at a cross-section of our small intestinal wall. On the outside, again, we've got that serosa, so that's our outer layer, makes up this part right here. Inside of that, we get the muscularis, so those are those layers here. Interesting, compared to the stomach, we only have 2 muscle layers here, longitudinal and circular, as opposed to the 3 muscles we see in the stomach. On top of that, we get our submucosa, so that's going to be our connective tissue that'll attach the muscularis to the much bigger mucosa layer, which is going to be responsible for the absorption that happens inside the small intestine. Now, the intestine is organized quite sophisticated, and one of the things that we're going to need to understand is the sophistication has everything to do with increasing surface area so that we can absorb more things from our food. So in the small intestine, we actually break down the mucosa. Remember, our mucosa on the last slide would be this whole layer here. We've broken that down into 3 other smaller layers. So we get the mucous epithelium, which is going to be the stuff right on the outside here. We get the lamina propria, which is the stuff we'll see in the middle in this area. And then we get the muscularis mucosae, which is this band that runs along here. That's going to be an extra layer of mucosa, a muscle that contributes to the mucosa. Okay, if we look at the very inside part of the small intestine, the very tip part of the mucosa, we're going to see that it's structured so that it has the maximum surface area for absorption. And the way it does that is it breaks it down into 3 different folds. The first fold we have is called the plique, which is this big finger-like projection. And on that plique, we're going to have all of these tiny little finger-likes and projections, and that's what we see right here. Those are called villi. On the very tip of the villi cells, we have the microvilli.